Okay, looks like we're recording now. So today what we're going to do is we're going to finish up our discussion of image contrast. We'll have a short breakout exercise where you can work on that together. Then we're going to talk about functional MRI and diffusion imaging. We probably won't get to phase contrast and geography, and so we don't. We'll pick that up on Monday. So first of all, just to remind you, last time we talked quite a bit about T1, T2, and T2 star, and the fact that these are sort of relaxation time constants that differ between tissues, and therefore we want to be able to take advantage of those differences in time constants to create image contrast. In addition, different tissues have different proton densities, so they have a different density of water, and so also want to be able to take advantage of that. And so by adjusting the pulse sequence, we can create contrast between different tissues. And so that's going to be the main subject of today's lecture in terms of image contrast. How do we take advantage of that image contrast? And we'll see that the two main ways of creating contrast are that we'll talk about today are by adjusting the echo time and the repetition time. Now, just as a reminder, and this was covered in the quiz, which most people did fairly well on. Those people who did the quiz sort of early on did very well, and sort of late answering responses seemed to be not so, sort of more diversity there. So remember, based on tumbling rate, we had that the T1 had this characteristic curve where it was sort of minimized at the Larmor frequency, and then the T2 had this sort of curve. Okay, so where it was maximized, it got bigger with faster tumbling rates and smaller for slower tumbling rates. And in addition, we also know that T1 is always greater than T2, which is always greater than T2 star. Okay, because T2 star includes both T2 and T2 prime, and so it's always going to have a faster decay rate or smaller time constant than T2. And similarly, T2 is affected by the same mechanisms that cause T1, and so it is also always going to be shorter than T1. All right. Okay, so we talked about the saturation recovery sequence, which will be the main sequence we're going to talk about for when we're talking about image contrast. And we said we could either have a gradient echo sequence where there was dependence on T2 star, or we could have a spin echo sequence and where we have dependence on T2. Right. The only difference in the pulse sequences here is in a spin echo pulse sequence, we have this 180-degree refocusing pulse. Okay. TE in both cases is where we go through the center of K space. So this would be when we had Kx equals 0. And so TE stands for echo time. And then TR is this distance between consecutive excitation pulses. Okay. So typically for a spin more pulse sequence, we'd apply 90-degree pulse. We acquire a line of K space during this regime. And then we wait some out till TR is done. And then we apply another 90-degree pulse, and then we'd acquire another line of K space. And so we go through K space line by line by line, TR by TR by TR. So that would be for a typical spin or pulse sequence. So what we want to do is we want to look at sort of the, take a closer look at this form here. So we have I of xy is equal to the proton density times the recovery, okay, T1 recovery times the T2 decay. So we're going to talk about three different types of images. There's going to be T1 weighted, there's going to be T2 weighted, and there's going to be proton density weighted. Okay, so those are the three types of image contrast we're going to look at first, and these are very common in the clinical regime. So for T2 weighted, we want to make the effects of T2 go away, so minimize T2 effects. Okay, and what we want to do is we essentially want to make this term, we'd like this term just to go to 1. All right, and the way we do that is by making TE as short as possible. Okay, so that would be a T1 weighted image. For T2, we want to, or sort of T2 weighted, we'd like to minimize T1 effects. So we'd like to make this term go to 1. And the way we do that is by making TR go to infinity, or making TR as large as possible. Okay. In general, we don't want to make it to go to infinity, but we want to make it long enough such that that term disappears or becomes 1. So this whole term, whoops, this whole term goes to 1, and then this term goes to 0. Okay, so this exponential term would go to 0, such that 1 minus that exponential term goes to 1. Right. And third, for proton density, we want to minimize both T1 and T2 effects. Right. And the way we do that then obviously is making TE go to 0 and TR go to infinity. All right, so that's the basic idea. And then beyond that, there's still going to be some selection of TE and TR, which we'll talk about next. But the general idea is these are some of the initial concepts that you want to have in mind is you're trying to get rid of either T2 or T1 weighting or both in these different types of contrast. So as we said before, T1 weighted is we make TE very short compared to either T2 or T2 star, and that will allow us to, so that T2 weighted term is gone, and we're just left with a proton density term and a T1 weighted term. All right. And so now the question comes, and now we want to pick a TR that maximizes contrast between two tissues, typically. All right. And here's the principle that if we have something with a short T1, it recovers very quickly. And if we have something with a long T1, it recovers not as quickly. And so there's going to be a difference in the signal intensity depending on what the TR is. Now, if we wait very long, then both species will recover. And so there's not going to be much difference. But if we wait the right amount of time, then there should be some contrast between the two types of tissues. So that's shown here, where we basically have plotted the, this is the 1 minus EXP minus TR over T1 term for two different tissues, one at 800 milliseconds and one at 1200 milliseconds. And so you can sort of see that the one with 800 milliseconds, this one recovers much more quickly or slightly more quickly than the one with 1200 milliseconds. Now, if we made TR very small, there's not going to be too much difference. And so this is the sort of difference between these two curves, so that there's not much difference here. Similarly, if we wait really long, both of the species have recovered to the equilibrium point. So we take the difference, that's also going to be rather small. So we don't want to be operating at those points. We see that the difference is maximized here, okay? And so we can see that the difference is maximized around 1000 milliseconds, okay? And so a good rule of thumb is if you have two species, you know, if you take the average, this T1A plus T1B, and average the two, then that's generally a good sort of rule of thumb for finding the TR that maximizes the contrast. Now in your homework, you're going to come up with a much more exact formula based on the T1s and T2 of what's the TR that maximizes the contrast. And just as a clue, just remember your calculus and remember how to maximize or minimize functions. Okay. But for some of the exercise we're going to do today, where we're not going to make you do very sophisticated calculations, picking something that's sort of the average of the two species that you really care about is a good rule of thumb. So T1 weighted scans are used quite a bit. For example, they're really used a lot in research imaging to look at the contrast between gray and white matter, and we'll see an example of that later. Clinically, one application of them is there are certain diseases that have sort of accumulation of fatty acids or triglycerides. And as you remember, fat has a rather short T1, so it will tend to be brighter than other tissues. And so, for example, in this case, where there's sort of an infiltration of the corpus callosum, you can sort of see that the fat appears very bright on this patient. And this is not something you'd see in a normal subject, right? So where we expect to see differences, or you want to accentuate differences based on the T1 of the tissue, then that's where T1 weighted imaging is very useful. For T2 weighted scans, basically we got rid of the T1 term, so we're just left with a proton density term and a term that's dependent on T2. And once again, now we want to try to maximize this contrast based on the T2. Okay, so we're going to pick a TE to maximize T2 contrast. All right. So we've gotten rid of the TR, we've gotten rid of the T1 because we've made the TR very long. Okay. So the question is, how long should you make the TR? On average, you want to make it 3 to 5 of the T1 of the species, of the longest species that you think is going to be present or that you care about, such that in 3 to 5 time constants, it is typically returned back to very close to its equilibrium value. So let's look at T2 contrast. So this is an example of T2 contrast where we have two T2 species, one at 80 milliseconds, which recovers more quickly than the species at 120 milliseconds. Once again, if we image too quickly, right, if we make the TE too small, then neither has decayed very much. So when we look at the difference, it's also going to be very small. On the other hand, if we wait too long, if we make TE really, really long, then both of them have gone to 0. And so once again, if we look at the difference, it's going to be very, very small. So once again, we see that there's sort of a sweet spot here. And in this case, we see that maybe TE around 100 milliseconds is good. And once again, as a rule of thumb, you could just take the average. Okay. And obviously, you could come up with a more exact expression if you wanted to by just taking the derivative of this function. But for rule of thumb, taking the average of the T2s is typically not a bad place to start. So this example of some T2 weighted images, you'll notice that in a normal brain, what you see has the brightest spots are these areas here, right? So this is very bright, this is very bright. You're seeing bright spots here. So all that is CSF. So that's all cerebral spinal fluid, which is the fluid that bathes your brain. And that has a very long T2. Remember, so it's basically, it's a fluid. And remember, it has fluids have very high tumbling rates. So they're going to have very high T2 because of the motional averaging. Right. So this is a normal brain. And so you expect to see the CSF. These are the ventricles here that you see in the middle of the brain. This person looks like a nice, young, healthy person. Typically, if you saw an older subject, those ventricles tend to enlarge with age and with certain diseases. And here, clearly in this subject here, there's something clearly going wrong here where there's essentially a cyst that's filled with fluid here. And so this person would clearly need some surgical intervention. And that's an example of fluid showing very bright on a T2 weighted image. Finally, the proton density images is we're going to make TR very long compared to T1, and we're also going to use a short TE. So we're going to get rid of both the T1 and the T2 effects. And so we're going to end up with something that's simply related to proton density. And this would be something where, for example, if you thought that the density of water was changing with a disease, that you might want to use that. And How do you know what to do? Oftentimes it's very observational. You have to look at a lot of images or look at subjects and you sort of see, what might be the case. In other cases, there might be some underlying mechanisms which would lead you to hypothesize that, okay, there's going to be more water in this situation than other, or there's going to be difference in T1 or T2. But a lot of it is based on observations and research studies. For example, this is an example where they found that when they looked at diffuse white matter, so all this area here, these areas here, this is all white matter, which we'll talk about later. That's sort of the wiring of your brain. And what they found out was there are areas of the brain and on a proton density weighted image where they did feel like there was a change of proton density. It's rather hard for me to see here, but they're claiming that they can sort of see deficiencies, I guess, in proton density in this subject with multiple sclerosis. Okay, so if there are no questions, what we're going to do is we're going to break into breakout rooms for this. And then what we're going to do is we're going to have, I've made Amanda and Vinay co-hosts. And so once we decide how many breakout rooms there are, we're going to then, we'll divide the breakout rooms and go through those. So let me explain the problem. And then you're just going to submit your answers through the chat, okay, for each group. So we assume we're using a saturation recovery sequence, and you are given 3 tissues, CSF, gray, and white. Okay, and just to tell you what is what, so in this image, that's the CSF. So the dark stuff here, CSF, the bright stuff here is CSF. So that's the cerebrospinal fluid. So it appears dark on a T1 weighted image and it appears bright on a T2 weighted image. The gray matter is, well, let's talk about the white matter first. The white matter is this stuff. It appears very white on the T1 weighted image. All right. And then the gray, so the white matter is the wiring of your brain. So you have a lot of wiring in your brain. And then the gray matter is sort of these gray parts that sort of are at the edges of the white matter here. So that's the gray matter, right? So for each of those, you're given a proton density where it's sort of scaled to CSF is 1 and the others have relative scaling. You're given the T1 of each tissue, and you're also given the T2 of each tissue. So for each of these images, the T1 weighted image, the density weighted image, and you're given examples here. Here's a T1 weighted image, density weighted image, T2 weighted image. You want to take your best guess at what value of TR and TE should you use to achieve these images. All right. So let's see, break into breakout rooms. Let's see how many. Okay, that's great. We're going to have 9 breakout rooms. And so I'll take breakout rooms 1 through 3. Amanda, can you take 4 through 6? And Vinay, you take 7 through 9?
Sounds good.
Okay, great. So we'll give you, let's say, about 5 to 7 minutes. Okay, let's take a look at the answers. I think most people are on the right track. So let's just take a closer look and see what we would want to do for this problem. So just make sure, can everyone hear me okay?
Yep.
Good, all right. So, and we are recording, okay. So let's look at the T1 weighted image. So we know that for T1 weighted, we want to make TE as small as possible. So you're not really given the constraint, so we might as well make it 0. So that's fine. In practice, for most clinical scanners, you could make it as short as 3 milliseconds. And for very, very special, what are called ultra short T, you can actually make it like on the order of 100 microseconds or 200 microseconds. So that would be fine. Then the only other choice is TR, right? And in this case, there's 3 different T1s to pick. But you notice 2 of them are fairly close together. And so we probably want to take the average of those 2. And those are spaced apart by what? 50, right? 500 milliseconds apart. So we had 250. So TR about 1100 would be good. Okay, so the average of the gray and white. And you can sort of see at that TR, because the T1 of CSF is so long, it's 4,000, CSF will still appear dark, which is what it does. See, CSF looks very dark. And so if we had a TR of 1100 milliseconds, that's not very much time for CSF to recover. So CSF will appear dark. So it makes sense that we spend most of use our TR to really maximize the contrast in these regions here at the interface between gray and white matter. So this T1 weighted image is really the bread and butter of a lot of research imaging. This is the type of sequence that is used to take a very nice anatomical picture of the brain and to sort of then, analyze the structure of the brain. All right, so that's for T1. For T2 weighted, we want to make TR go to infinity, but clearly we don't want to wait infinity because that's too long. So what we want to do is make TR roughly 3 to 5 times the longest time constant. So the longest time constant is 4,000. So in this case, we want to make TR, anywhere from 12 seconds to 20 seconds. Okay. So, in general, at 12 seconds, you're already at 95% of the recovery, so that would be fine for most cases. All right. But anything on that order would be good. And the TE, once again, we want to sort of take, you notice of these 3 T2s, 2 of them are close together and the other one is way out there. All right. So we want to take the average of the 2 that are close together. And so that would be 95 milliseconds. And notice with 95 milliseconds, 95 is very small compared to 2000. So, the CSF has hardly decayed in that time. And so you sort of see that the CSF is very bright with that. And then we're sort of maximizing the contrast between gray and white matter again. All right. And then finally, for the density weighted image, we would want to pick T2, once again, close to 0, and TR, let's say around 12 seconds or so. All right, so that's sort of basically what you want to do, and it looks like most people were on the right track there. So are there any questions before we move on from this example?
Professor?
Yes.
Can you remind, what is the density weighted image?
The density weighted image is one where you've minimized both T1 and T2 weighted image such that your signal, the image you get, is roughly proportional to the density of water or of hydrogen protons. Okay, so this is really an image of, this density weighted image is basically saying, in the CSF, in the ventricles, I have a lot of water because that's mostly water. In the white matter, the wiring tends to have less water in it. And then the gray matter, which has all the neurons in it, tends to have more water than white matter, but less water than the cerebrospinal fluid.
Thank you.
Sure. So on your homework will be posted later today, there is, so the question is, what is the TR for proton weighted? It's about, this is about 12 seconds. Or 12,000 milliseconds. Okay, sorry about my handwriting there. All right, and so for the homework, there's gonna be a MATLAB exercise where you're sort of given some images and you're supposed to sort of simulate creating a T1 weighted, density weighted, and T2 weighted image by sort of weighting the different tissues by, that equation, the saturation recovery equation. All right, so if you apply these principles to that problem, it should be fairly straightforward. Okay, any other questions before we move on? Okay, so I'm going to talk about 2 other types of sequences. One is, so far we talked about saturation recovery, which means we have a 90-degree flip angle, and then we wait for the magnetization to recover. And as you can sort of see that, we're spending a lot of time waiting for the magnetization to recover before we can have enough magnetization to use again. And so one of the insights that was made very early in the development of MRI was, well, instead of flipping the magnetization all the way to 90, why don't we use a smaller flip angle such that, for example, maybe we don't get as much magnetization to work with, but we don't have to wait so long for it to recover again. So for example, if I used a flip angle of 30 degrees, right, I get half the magnetization flipped onto the MXY axis, and then I would still have square root of 3/2 of the magnetization left. And so I don't really have to wait for it to recover so long. And so this has led to these sort of flash sequence, which sort of stands for low flip angle kinds of sequence. And so the major innovation here is simply that we make the flip angle less than 90 degrees. And by doing that, it turns out we can make the TR very, very short. And that allows us to have very rapid imaging. And so flash sequence is also very commonly used, especially where speed is of interest. So typically, for example, when you have an MR exam, the first exam you have is what's called a localizer exam. And that is just trying to get very quick images of where the brain or the body is located, such that then the remainder of the exam can be prescribed and the slices can be put onto those localizer images. So the intuition behind this is similar to what you might have if you had a flush toilet, okay? So if you have a toilet with a water tank in it and the water is all the way filled up in the tank, and if you flush all the water, you'll get a very strong flush, right? But then you have to wait a long time for the water to refill again before you can flush again, okay? So there's the pro that you have a very strong flush, but there's the con, which is where you have to wait a long time. So those of you who've sort of gone through water conservation and stuff know that sometimes now there are these low flush toilets, or even some people have put bricks in their toilet tanks to make the flush less. So the idea is, instead of doing a full flush, what if you just did half a flush, right? You could just use half the water or maybe a third of the water, still get a reasonable flush, and now you don't have to wait so long for the water to accumulate back before you can do your next flush. So that's sort of the intuition behind flash, that you're getting a reasonable amount of magnetization just by using a smaller flip angle, and then you don't have to wait so long for it to recover, right? So these are some examples here where, for example, it turns out for a given TR and T1, there's an optimal angle called the Ernst angle, which is where the signal intensity is maximized. So all these maximum occur at what is called the Ernst angle. So this is the Ernst angle. And you can sort of see here, for example, for a TR of 500 milliseconds and a T1 of 1,000 milliseconds, if I use the Ernst angle, which in this case is around, let's say, 52 degrees, I still get half the magnetization or half the signal intensity that I would get if I had a 90-degree pulse. And for example, for a 90-degree pulse, if I had to wait 3 T1s, let's say I had to wait 3 times T1, then that would be about 3,000 milliseconds, right? So to wait for things to recover. Whereas in this case, I'm only waiting 500 milliseconds between lines. And so with this type of sequence, you can go much faster. And so you'll see a lot of these types of sequences in clinical use and even research use, just because speed is always an issue when we're doing MRI or any type of clinical exam. The other type of sequence that's very important is what's called inversion recovery. So we're going to talk about that a little bit. So we've looked at this before. This is just the spin echo part of the sequence, right? Now what we're going to add is what's called a preparation pulse, or also an inversion pulse, okay? And all this does is it takes the spins and 180 degrees, if the spin is pointing up, 180 degrees will just flip it down, okay? So you're gonna go from magnetization that's at M0 to minus M0, and then it's gonna recover back like that, right? So where the intensity then depends on where you acquire the image. And so typically after we apply the inversion pulse, we acquire the image at some time Ti, which is called the inversion time. And the nice thing about this is, let's say this is 0. With the correct inversion time, you can actually make it so that if you acquire at that time, the magnetization is 0. And so you can null out a species. And in fact, given a TR and a T1, you can pick the inversion time that will give you an intensity of 0. And so this is a nice way of essentially erasing out tissue types that you may not care about, okay? So this is an example of an inversion recovery sequence, where in this case, showing the different inversion recovery of fat, which recovers more quickly than white matter, which recovers more quickly than CSF. So by acquired at this point, I would null out fat, okay? If I null, at this point, I null out white matter, okay? So in here, here I've nulled out fat, here I've nulled out white matter. So you can sort of see here, all that white matter is dark. So white matter is 0 here. And then here, at this point, I've nulled out the gray matter. So the gray matter here is almost 0. And at this point here, the CSF is essentially 0, okay? So just by acquiring at different points after the inversion pulse, I can erase different parts of different types of tissue. That's just shown here. For example, here I nulled out fat. Here I would null out fluid, and here I will null out blood in this case, right? So here's some examples of inversion recovery. Oftentimes, as we saw, there can be a lot of signal from CSF. So it's very common to try to null out CSF, and such that, you know, if we're looking for something that's not CSF, and we don't really care about the fluid or edema or those types of effects, this can be very useful for nulling out CSF. The other thing is, this is sort of like a black blood sequence, where in order to see the vessel wall or the cardiac wall, we want to try to get rid of the effects of blood flowing, and then this allows us to basically null out the signal from blood such that we can better see the tissue in the cardiac wall. And finally, this is a sequence that's very much related to what you did on one of the homeworks, which is you can have something called a background suppression pulse, where you can actually null out, this is over a range of T1s, you can null out a lot of things, and by applying a lot of these 180-degree pulses, you can sort of erase a lot of the background. And so this is an example here where, for example, let's say all you cared about was the blood flowing into a region. You can null out all the tissue, all the muscle, all the fat across a range of T1s, such that all you get is an image of the blood that's flowing in, and you're not seeing the background tissue, right? So that's another method that's actually used quite a bit as well. Okay, so if there are no more questions, we're going to switch gear a little bit, and we're going to switch to functional MRI. So are there any questions on image contrast before we switch to functional MRI? Okay, so let me just see where things are here. Okay. Okay, so up to now, and especially in terms of talking about contrast, we mostly focused on image anatomy, brain anatomy. And so if you're applying MRI to the brain, then MRI initially was only used for brain anatomy. But over the last, say, 30 years, there's been an area called functional MRI that has arisen, which allows you to use the MRI system to study brain function. So we're going to talk a little bit about how that works. So the setup is very similar to an MRI exam, in which case, you know, you typically just put the person in the scanner. The only difference with the fMRI exam is in a lot of the experiments, you're actually having the person actively doing something, maybe playing a video game, maybe gambling, maybe watching scary pictures, maybe you know, looking at different political ads. You're basically having them do something, and then you're watching what their brain does as they're doing it. So fMRI is a relatively recent phenomenon in terms of discovery. This is Seiji Ogawa, who was really the sort of the pioneer of fMRI. And this was a paper back. So it's really the early 90s where the first measurements first came out. And this was from one of their papers where they essentially just shined a light. They turned it on and off. And here they're looking at the MRI signal in this part of the brain. The back of your brain is where your visual cortex is. And so they were looking at sort of in one area of the brain, you know, how the signal is going up and down. Okay, there was a question. Are the images where you just see the vessels ever used live during surgeries? That's a good question. I mean, we will in a later lecture, those images, those particular type of background suppression is probably not the most popular way to look at vessels. I mean, there are, you know, and I'm not sure that they're actually used live, but clearly, definitely for pre-surgical planning, we will acquire what are called angiograms or MR angiography, which we'll talk about probably on Monday, where you can actually see what the vessels look like and see where the occlusions might be. So those would probably be used both pre and post surgery. During surgery, you basically can see the vessel anyways if you open up, and so it's probably less frequently used in that capacity. But there are interventional things where they might have a surgical bed where they take the subject over to the surgery, and then they have a rail and they slide them over and put them in an MRI scanner and move them back and forth. And that has been used. The problem with using MRI during surgery is that it's just really, sort of the form factor makes it a little more difficult to do it, especially, if you have this big tube. Are there downsides to suppressing certain tissues rather than segmenting separately? There's always downsides to any method, and typically where suppression is very useful is where you're really, the background tissue might have a lot of information that you just don't care about, and so in that sense, as long as you can suppress it and you don't really care about it, then there's really no downside to suppression, and it also improves sort of your signal to noise ratio. But so that's really if you really want to look at vessels on their own. If you want to segment them, is that because the preamps and MRI changed their gain? The preamps, no, it's more because of just, if you are looking at, typically blood signal can be very small compared to the background. And so if you get rid of the background, then you can actually, anything that perturbs the background is causing noise. And so you're sort of reducing that noise factor as well. Also, segmentation is not that easy to do. I mean, even after you suppress the signals in angiography, even the segmentation can still be difficult to do. So suppression just makes that easier. Okay, so getting back to fMRI, so what we're seeing here is the response of the brain to light shining on and off. And one key thing to realize is we've been talking a lot about TEs. Here they use an echo time of 40 milliseconds. You can sort of see a fairly nice, robust response. When something is first discovered, it's really, once it's discovered, it's sort of obvious. But when you're in the early stage of discovery, it's really not clear what's going on. And so one of the things they did was they looked at different echo times. So here they did an experiment at a shorter echo time. And you can sort of see here that at a shorter echo time, there's almost no signal going on. So then that means that this must be related to something that's affecting T2 or T2 star, okay? This is just showing, how, there's just hundreds of thousands of publications in fMRI, very popular field. One of the trade-offs we make is that to acquire a very nice high resolution image, say 1 millimeter, isotropic voxels, typically you'll spend about 6 minutes to acquire a really nice image. But the problem with looking at brain function is in 6 minutes, your brain does a lot of different things, right? I mean, 6 minutes ago, we were doing something completely different, right? So we will make a trade-off typically in fMRI. This voxel, with typical voxel volumes, now it's much better. Now we're down to about 2 millimeter isotropic. So since this slide was done, I should update this slide. We're only at 8 millimeter cube. Okay, so we're at fairly good resolution now for whole brain. And then the advantage is now for a whole brain, we can acquire whole brain 8 millimeter cubic in about 2 seconds. Or even as short as, let's say 1 second actually is state of the art. So that's pretty good. That means every second you can get basically a picture of what the brain is doing. And so that's the trade-off functional MRI makes. It uses advanced techniques to go through the brain very quickly such that we can get these fairly good snapshots of what the brain is doing. The bread and butter of fMRI is echoplanar imaging, which is basically this zigzag trajectory that you saw in one of your homeworks. Basically, instead of going line by line, after each RF pulse, we just try to zigzag through K-space as quickly as possible. Now, there's many tricks that have been developed over the years to do that efficiently, but this is the basic idea. And on many research studies, this is still the bread and butter. So what does an MRI experiment look like? So remember, we talked about MRI, we're just looking at anatomy, and typically there's just going to be one image that you're going to look at. In fMRI, we actually acquire multiple images over time. And typically while we're doing that, we're also maybe having some stimulus, like say something that's off, and then we're having a checkerboard, let's say flashing during the on period. Okay. And we basically acquire images throughout that experiment. And we typically will repeat that. And so in here case, we're having the checkerboard on during these periods of time. Okay. And so we're looking for brain regions where there are signals going up in a fashion that's similar to the stimulus that we applied. Right. So the most basic type of analysis you could do is you could simply take your stimulus function, you could correlate it with the signal in every voxel. And if you did that, you can end up with like a correlation map. And showing here in this case, it's really the visual cortex that's lighting up. Okay, and so this is what's sort of called correlation analysis for fMRI, and it's still used quite a bit, although there's much more sophisticated methods, but this is the basic sort of way to think about it. So in this case, for example, the person was tapping their finger, and this is showing what area of the brain, essentially in their primary motor cortex, that's lighting up as they're doing this task. So let's talk a little bit about what the mechanism is. And remember, this was the some slides from the original image. And once again, emphasizing that when we had a longer echo time of 40 milliseconds, we got a nice signal. And when we had a shorter echo time of 8 milliseconds, we had a much shorter, much reduced signal. So it turns out that the thing that we really have to revisit is R2 star decay. So R2 star is just, well, it's right here. R2 star is just the inverse of T2 star, which we've talked about. The reason we talk about R2 star is a little more related to the physiology of what's going on. And so that's why, the term R2 star is used a lot. So instead of, e to the minus TE over T2 star, we can talk about e to the minus R2 star times TE. Right? It's the same thing, it's just a different, we're talking about relaxation rate instead of relaxation time constant. And remember, R2 star is always composed of two components. One is the sort of the T2 component, which is the random motion of spins, and one is the T2 prime, which is due to static inhomogeneities. In fMRI, we primarily rely on T2 prime. There are some people trying to use the T2 effects, but the effects tend to be much smaller. So by and large, most people focus on the T2 prime effects, right? The basic idea is what we talked about before when we talked about magnetic homogeneities, in the sense that if in the presence of sort of a mild homogeneity, so this is mild homogeneity, different spins are in different locations. So let's say these are spins around a blood vessel. The fields are a little different around that blood vessel in different positions. And so the spins will get out of phase. And then at some point, if we come and measure at echo time, the overall summation of those magnetizations will have decreased. Right? Now there's much more homogeneity around that vessel, then there's going to be much more dephasing of the signal, much bigger decrease in the MR signal. So what is the bold signal change? The bold signal change is really due to changes in the R2 star. Okay, so we typically have a baseline signal, which is some equilibrium magnetization and EXP minus TE times R2 star B, where this B stands for baseline. So we're going to have some decay during the baseline. And then when the brain is active, typically what we see is this phenomenon. Remember, we saw the MR signal go up with activation. So the signal is going to go from here to here, right? So we're going to have less, R2 star is going to decrease, right? And so this signal in the activation is going to decay much more slowly than it did during the baseline. And typically what we look is at the bold sort of relative change or the percent change. So that's equal to the activation value minus the baseline divided by baseline. And it turns out that's roughly proportional to minus TE times delta R2 star. And typically for most experiments, delta R2 star is less than 0. Okay, so this whole term is we typically see a positive bold change. Now that's not always the case, but the most prevalent change we typically see is a positive bold change. So because that relaxation rate with activation tends to decrease such that delta R2 star is less than 0. Usually. Okay. So let's talk a little bit about what changes R2 star. So R2 star depends on deoxyhemoglobin. So what happens is, you know, when the deoxyhemoglobin molecules go into the lungs, let's say in the lungs, they bind to oxygen, so we just shown here in red. And so essentially, they carry oxygen into your brain. And then when they go to the brain, the oxygen offloads from the deoxyhemoglobin molecules. And so what this exposes is the iron at the core. And so that iron acts to perturb the magnetic field around that. So the more oxygen that's offloaded, the more iron is going to be exposed, and the more inhomogeneity there's going to be, and therefore more dephasing. So the picture is this, that in some, let's say we have a condition where there's some of the iron is exposed, but not too much. So there's some moderate dephasing of the spins. So there's some deoxyhemoglobin, and so some dephasing. Now, if there was a case where a lot more oxygen became offloaded, then much more of the iron would be exposed. And so there'd be more deoxyhemoglobin, more dephasing, a decrease in the MR signal, higher R2 star. Okay, so that's the basic physical picture to have in mind in terms of what's going on with fMRI, that it's really related to how much deoxyhemoglobin is there. And so the amount of deoxyhemoglobin that's there, it turns out to be this sort of trade-off between blood flow and oxygen metabolism. Okay, so blood flow is coming in and delivering oxygen, right? And then metabolism is using up the oxygen. So there's this competing factor, all right? So blood flow coming in gives you more oxygen, right? If there's more blood flow, there's going to be less deoxyhemoglobin, right? Whereas if there's more metabolism, then a lot more of the oxygen is going to be used. So that's going to tend to increase the deoxyhemoglobin. So there's going to be this sort of competing factors of one mechanism is trying to deliver oxygen, the other is trying to use it up. And so the question is, which one wins out? Okay. If they were equally balanced, there'd be no change in deoxyhemoglobin. There would be no fMRI signal. If we used up more oxygen than we delivered, then the overall deoxyhemoglobin would go up, and we'd expect a negative bold signal. In fact, what happens is nature has set it up such that the percent change in CBF over the percent change in CMR2 is typically around 2 to 3. So that means for sort of every percent increase in metabolism, the brain has set it up such that it's going to deliver roughly twice as much an increase in blood to really make sure you have enough oxygen to do what you need to do. This is especially true for areas like the primary visual cortex and your motor cortex. Essentially, you know, if you're in the jungle and you're being chased by a bear or a tiger, the brain is set up such that you better have enough oxygen to keep going, because otherwise, you know, you're not going to survive. All right. So there is this coupling ratio that turns out to be around 2 to 3, and because it's greater than 1, that gives rise to the fMRI signal. This is a cartoon that one of our grad students made up many years ago, sort of trying to capture or summarize what's going on. So we have CBF delivering oxygen and CMR2 using oxygen. So red here is oxygenated oxyhemoglobin, and then the purple is sort of representing the deoxyhemoglobin. So typically what happens is, let's say there's some increase in neural activity, there will be oftentimes a very sudden increase in metabolism. And that's going to increase the amount of deoxyhemoglobin, and that there's more deoxyhemoglobin, so there's going to be more dephasing, so that the MR signal will go down. This is what's called the initial dip, and you can sometimes see it. It is a real thing, but it only happens very instantaneously. So it's right at the point where the metabolism increases before the blood flow has had a chance to sort of do its work. But eventually the blood flow increases quite a bit, sort of 2-to-1 ratio to the CMR2. And so you have a lot of oxyhemoglobin now, and therefore there's less deoxyhemoglobin overall. And so therefore there's less dephasing, and therefore the MRI signal will go up. After it's all done, things return back to baseline. And how that happens is still actually an active area of research, but at some point it does return. Okay, so let's just to check in and make sure people are on board with that. Let's just do a couple of polls that are, make sure we have the basic concepts down. So let's do the first question here. So the magnitude of the bold signal percent change will either increase or decrease as a function of the echo time. So go ahead and put in your answer there. Okay, about another 10 seconds and then we'll end. Okay, let's end the poll and let's take a look at so actually fairly close here. So let's remember the bold percent signal change, right, is, it's the difference between something like this and something like this, right? So it is going to have sort of this, the difference is going to look like that. Okay, if we just took the this was the SA and this was the SB and this was SA minus SB, right? But then if we the percent change, we have to divide by SB. So let's say we divide it by SB, then that's going to actually, the percent change SA minus SB over SB is proportional to minus delta R2 star times echo time. So in general, that is going to increase, that percent change is going to increase with echo time. And so in this case, increase would be the better answer. And now, how do we reconcile with the fact that there does seem to be this optimal echo time where the difference is the greatest? So this is the absolute difference, SA minus SB. But remember, if we divide by SB, SB is getting smaller with time, such that that overall thing will tend to also increase. Okay, so the percent change will increase. Now the problem is, this is sort of a trick question because you can't make echo time too big because at some point there's no signal left. So it's almost like you're taking 0 over 0. So even though 0 over 0 is a big number, it doesn't really make sense. Okay, so in the sweet spot where you might be measuring, increasing echo time does help and makes the bold signal bigger, but after some point, it doesn't really help anymore. Okay. Okay, let's do one more of these questions. Let's do question 2. So an increase in the functional CVF response will tend to either increase or decrease the bold signal. Okay, about 15 more seconds. Okay, let's take a look here. So here are the results. So yeah, so about 2/3 said increase, and so that is the correct answer. It will tend to increase the signal. Remember, if we have more blood flow coming in, okay, then there's gonna be more oxygen coming in, right? And so that means that there's gonna be less dephasing of the signal, so the MR signal should go up, all right? So that's one way to think about that. So we might, in the interest of time, we're going to skip over the next couple of questions and just go to sort of more of the fun applications of fMRI, and then we'll pick up on diffusion next time. So, you know, I think when Matt Groening did this cartoon, you know, maybe it was a bit in jest, but it does turn out that pretty much everything he talked about, you can sort of find, you know, a paper or some study. Now, when I did this slide, I mean, I couldn't find anything that was specifically donuts, but there certainly is a lot on, you know, studies related to, you know, obesity and different types of, you know, sort of eating effects. Okay. I think one of the definitely one of my more favorite fMRI experiments is this one here, where this was done at Caltech, where the experiment was basically, in some cases, you're basically looking at wine, and in some cases, you're given the same wine, but in certain cases, you're told that the wine costs $90, and let's say in other cases, you're told it costs $10, and you're asked to rate how much you like it. And even though it's the same wine, if you're told that the wine costs $90, you like it way more than if it's $10. Similarly, if you're told that it's $45, you like it way more than if you're told that it only costs $5. Right? So for whatever reason, you know, what you're told as the price is signaling to your brain and affecting your pleasure or your liking of it. And so what they did was they essentially put people in the scanner and looked at their fMRI response. And you can sort of see here, and these are in sort of the reward centers of the brain that sort of you know, monitor how much you like something. So here is $45 wine versus $5 wine. Okay, here's $90 wine versus $10 wine. In both cases, not only is the you know, the initial positive response much less, but it does seem that there's almost sort of this negative response afterwards. We're not sure exactly what that means, but it certainly is showing that your subjective sort of liking of something is also, there is a biological substrate to that. There is, for example, using fMRI now, we are just at the initial stages of being able to read people's minds. And so this was a, I think I can play this here. So what you're seeing here is they played a movie on one hand, and then they put the person in the MRI scanner, and then just by looking at the signals in the brain, they tried to recreate what that image was simply by looking at fMRI signals. And you can sort of see here, it's not perfect, but it's doing a pretty good job, you know, recreating, in this case, Steve Martin, I think is Inspector Clouseau in the Pink Panther. You know, for certain things, you know, I think that's pretty good there. And so we are just at the early stages now of, you know, being able to sort of use the fMRI to sort of get a better sense of what's going on in the brain in real time. So we're going to end there for now. We'll finish up fMRI next time. And what I'll do is I'll stay around here for office hours. If there's any question on the homework, or yeah, the homework is still, it's not due till Friday. And also, if people have questions on their projects, you can certainly ask them during office hours. All right. So otherwise, we will see you next Monday then.